Welcome back to another edition of the GWU Dermatology Translational Science Lecture Series. I'd first like to thank our sponsors, Celgene and Pfizer, for affording us the opportunity to bring a wonderful speaker to us today. Dr. John Zippin is a longtime friend and colleague. He's assistant professor of dermatology at Weill, Cornell. He is both the quintessential physician scientist, but also, I'd say, very much a modern day physician scientist, having a very busy clinical practice focusing on contact dermatitis, but also having both an NIH and industry-sponsored lab, doing some of the most integral work in the world of cyclic AMP biology. In fact, I'd probably argue he is the foremost world's authority of cyclic AMP as it relates to skin disease. But I would have to say, of all his accomplishments, papers, textbooks, mentoring students, his probably greatest achievement would be the Zippin MD PhD, the world's oldest roller coaster. Actually, it was originally called Pippin. Somehow he stuck his name in there. So
no relationship.
I'm sure don't deny. I know you're bashful.
No, I know it only because anytime anybody Googles me, they always ask, like.
Follow-up question: why are people always Googling? So, on that note, why don't you come up? And we're so fortunate to have Dr. Zippin with us today.
Trust me, it will happen after. You Googled. That's how you found that. Well, it's really a pleasure to be here. I always like coming to DC. It's a good experience overall to be near the nation's capital, but also it's fun to see Adam. I miss him when he left New York. It was really it was a sad day. So I actually met Adam when we were both residents. There are all these resident competitions for those who are interested in science, and for those of you who don't know, Adam's really done amazing work on nanoparticles, and so we met this first time at this New York Academy of Medicine competition, where I won first, Adam won second. only, and the friendship blossomed, and only after the second competition where he won first and I won second did I realize that I really needed to step up my game. And this went back and forth over the many resident competitions throughout our residency. And then, the old saying is, keep your enemies close. You keep your friends close, your enemies closer. So we decided that we would really take our relationship to the next level and study together for the boards. And I was lucky when I started my faculty position at Cornell, I was given an office space. So we sort of hid out in that space and studied for, it must have been like 6 weeks or so, every day, to the point where my wife was like, "Where are you going every day? Who are you with? What are you doing?" Which is, how she eventually met Adam. But since then, it's really been it's really been great to know him, and you guys should all be very honored and pleased to have him as your program director, because there's very few people who can both teach dermatology, lecture about dermatology in a way that really keeps you. Keeps your attention. So I'm really happy to see him speak. Okay. So all joking and roller coasters aside, let me tell you a little bit about who I am and what I do. So, as Adam already mentioned, I am the director of the Occupational and Contact Dermatitis Unit at Cornell. We do hundreds of patch tests a year, photo patch testing, general standard patch testing, photosensitivity testing, working up for people like porphyria, et cetera. But that's sort of half of my life, or it's really a quarter of my life. The other 2/3 of my life, I'm in the laboratory. And what I've been working on for probably almost 20 years now is understanding cyclic AMP biology in skin diseases. And so the lab predominantly works on pigmentation, understanding melanocyte biology, and inflammatory diseases. So a little bit of melanoma, a little bit of pigmentation, and a little bit of inflammatory skin disease, all focused on understanding cyclic AMP. So some disclosures. Sorry, you guys in the back, you're part of my disclosures. I'm a founder of a company focused on using antibodies against cyclic AMP signaling proteins in diagnostics for melanoma. I'm also part of a company that has developed a wearable UV sensor. And then I received support from Celgene and Pfizer to investigate the role of cyclic AMP in inflammatory skin diseases, as well as from Metametrics. Okay. I always hate it when it does that. All right, so I think to start this whole lecture off, and I'm sorry, I tend to look left. It's not that I don't like the people on the right, but just tend to turn left. This is my good side. But I'll make a concerted effort because I do want to, if anybody has questions along the way, I do ask that you raise your hand and we stop. The lecture's not meant to like push the hour. And I know there's usually some questions that come up, especially understanding how cyclic AMP signals. And I think it's really important to understand how it works, to understand how the drugs that we're going to talk about are thought to function. So if we're going to talk about cyclic AMP signaling, we got to talk about what everyone's probably learned in the textbooks, which I'm going to introduce you to in the beginning and then tell you why it's incomplete and in some cases even wrong. But it's just the way it's been taught because it's a dogma in the field for the last 30 years. And the way you've probably been taught about cyclic AMP signaling is you have a ligand. If we're talking about a melanocyte, this would be MSH, binding to a G protein coupled receptor, which would be the MC1R receptor. That activates a heterotrimeric G protein. GS alpha is the business end of that molecule as it relates to cyclic AMP. And then that heterotrimeric G protein binds to a class of enzymes we call TMACs, or transmembrane adenylyl cyclases. They're called transmembrane adenylyl cyclases because they have these 12 membrane-spanning domains, and that sits in the plasma membrane, and that's what keeps it there. It only signals at the plasma membrane. It doesn't signal anywhere else. Maybe in recycling endosomes, but essentially it's associated with the plasma membrane. And when GS-alpha binds to the TMAC, ATP is converted to cyclic AMP. Cyclic AMP is a freely diffusible small molecule, so this is able to move throughout the cell, and then it can bind to effector proteins. Now, the effector protein that most people know is protein kinase A, or PKA. PKA is a heterotetramer. It's composed of 2 regulatory subunits and 2 catalytic subunits. When cyclic AMP binds to the regulatory subunit, leads to a conformational change and allows the catalytic subunit to then be free to phosphorylate proteins that are nearby. Now, about 20 years ago, there was a class of proteins discovered by John Scott called ACAPs, or A kinase anchoring proteins. What these proteins do is they anchor PKA to different places throughout the cell. And there are, of course, ACAPs that anchor PKA right next to those TMACs I just introduced to you at the plasma membrane. But there are other ACAPs throughout the cell. There are hundreds and hundreds of these ACAPs. And these ACAPs anchor PKA to these different areas throughout the cell. It could be at the mitochondria, inside the nucleus, on the cytoskeleton. And so this created a conundrum in the field, and this is sort of where I started my work in cyclic AMP. And the conundrum was, how can cyclic AMP reach these more distant sites? People thought that there might be diffusion of cyclic AMP, but in order to create a diffusive force strong enough to reach these more distant sites, you'd have to consume so much ATP, it just didn't make sense. So there had to be a way that you would reach these more distant sites. You further complicate the picture by introducing the topic of the lecture, phosphodiesterases, or PDEs. PDEs function to catabolize cyclic AMP or cyclic GMP into the inactive molecules AMP and GMP. These phosphodiesterases are present throughout the cytoplasm, and so in effect they prevent diffusion of cyclic AMP. So it became very challenging to understand how this diffusion could be taking place. To summarize about 20 years of work, there's been a lot of effort done in using techniques called FRET. FRET is a way to measure the amount of particular molecules or the phosphorylation or changing of proteins, and there are FRET sensors that can measure how much cyclic AMP is in a specific location, and these sensors have been used to show that cyclic AMP in fact does not diffuse great distances. It's actually very, very, very well localized to specific areas where the cyclases are. Okay, so then people would say, well, the protein kinase A can be activated here, and that diffuses throughout the cell. Well, literally, it was just like hot off the presses. People in the field had been talking about how that doesn't make any sense. If you have a kinase that's floating throughout the cell, it's going to phosphorylate all these different substrates. PKA is an incredibly good kinase, and you would lose all the specificity and selectivity of signaling. Well, John Scott, same guy who discovered ACAPs, recently published a paper which very nicely showed that PKA does not diffuse at all either. When PKA is activated, the catalytic subunit phosphorylates only the proteins that are in this small domain next to the activated protein kinase A. So you have to have a system now where, in order, at least the PKA is concerned, where cyclic AMP is made in little pockets throughout the cell. And it gets even more complicated because there are other effector proteins. There's channels. There's also this class of protein called EPAC, exchange protein activated by cyclic AMP, which regulates the small G protein RAP and leads to phosphorylation. Has a lot of cancer implications, other signaling implications. So to fast forward a little, one explanation for how these different domains of cyclic AMP could be functioning was to have another source of cyclic AMP. And most of my work has been on the discovery and establishment of a soluble adenylyl cyclase, which is actually more anciently conserved. It's conserved all the way back to cyanobacteria, whereas the TMACs show up in dictyostelium. But I'm not going to focus on this enzyme. This is more just to show everyone that cyclic AMP is made in all these different places with their associated phosphodiesterases, preventing the diffusion of that cyclic AMP. Why is that important? It's important because just because cyclic AMP can be made in all these different locations, do not assume that cyclic AMP does the same thing. Most of the work that we have published and others who have followed on our footsteps have shown that cyclic AMP made from the TMAC versus cyclic AMP made from the soluble AC in the same cell, depending on the signal, can have drastically different effects. One cyclase can make a neuron cell go left, whereas the other cyclase makes it go right. In beta cells of the pancreas, we know that this enzyme is responsible for amplification of insulin release in response to GLP-1, glucagon, whereas the soluble AC senses, it's an intrinsic metabolic sensor, senses glucose and leads to cyclic AMP that's required for any insulin release. So these are just a couple of examples of how understanding how these microdomains are functioning can help us to understand how cyclic AMP being elevated in a particular area is important. It's also important to realize that a phosphodiesterase inhibitor, which we all know raises cyclic AMP, cannot function unless you have a functioning cyclase. So if you think about it, if the cyclase isn't working, if I block the PDE, it has no effect because the effect is due to elevated cyclic AMP. So you must understand what cyclase is functioning in addition to understand what PDE is associated with it to truly understand how cyclic AMP signaling is able to affect disease and how it's normally functioning in the body. And that's why the majority of my work is on understanding the roles of the cyclases in the context of the different PDEs. Okay, so the lecture is going to be focused on phosphodiesterases. So let's talk a little bit about what we think we know about how phosphodiesterases control cyclic AMP in the cell. Now, all of these slides are assuming that the cyclase is functioning, it's on, it's doing its thing, and now we're just thinking about the phosphodiesterases. So in example A, we have phosphodiesterases as a diffusion barrier, and this is essentially the most widely held model of how PDEs control cyclic AMP signaling. So you have your cyclase making cyclic AMP, which is depicted as the red dots, and then you have the little Pac-Man, the little PDEs that are chewing it up, and that's preventing this cyclic AMP from reaching other areas of the cell. So you think of it like a dam, right? Like the Hoover Dam. It's preventing the flow of cyclic AMP to other areas of the cell, right? And all the FRET-based experiments have shown this to be true. And we know that in those FRET-based experiments, if we block the particular PDE, you start to see cyclic AMP showing up in other areas of the cell. So we know that A is definitely true. And we also have the model predicted in B, which we don't have any evidence to say for or against at this point. But in this particular model, the phosphodiesterases are sitting in domains throughout the cell where they're protecting that domain from cyclic AMP getting too high. And so when they are inhibited, whatever effector protein is sitting here is under normal conditions is not active. When these are inhibited, you would have then elevation of cyclic AMP in this white area, and you would have the signaling from that cascade. And then you have a more complicated, so the way I think about this is sort of the guy with the leak in his boat, and he's got a bucket, and he's throwing that water out as fast as he can. And if we gave him a smaller bucket by inhibiting the bucket, sort of inhibiting the PDE, or if he took his bucket away, his boat would sink. And that's sort of the model I think of in B. Now in C, this is a little bit more complicated, and I think this doesn't work unless you think of different PDEs. So there's many different isoforms of PDEs, which we'll get into in a second. I think that this particular model, you have different PDEs functioning in the same cell, and you're diverting cyclic AMP to different areas of the cell. There's less evidence to really support C. It's very hard to really nail down something like this occurring. And I guess the way you would think about this, the way I think about it, is a rushing river, and the PDEs are rocks in the river that are preventing the water from getting to those specific areas. And how is all this possible? How can we think of PDEs doing so many different things in a cell? Well, because there's many different PDEs. This is actually an incomplete list. I just happen to like this figure because it gives a lot of detail. There's actually PDE 10 and 11. But PDE 10 and 11 are bifunctional. They don't really hit cyclic AMP specifically. So we'll talk about each of these in some detail right now. So PDE 1 is a particular PDE that is regulated by calcium. So that's these CAM kinase and calcium binding domains. Also, it's a PK phosphorylation site. This is the domain that degrades cyclic AMP. And you can see that it has a really bad KM, almost 100 micromolar cyclic AMP. So it's not a very effective phosphodiesterase under physiological conditions. PDE2, about 30 micromolar. This has different targeting domains. It's regulated by other proteins as well. PDE3, again, these are all bifunctional. They hit cyclic GMP and cyclic AMP. And then you have PDE4. That's the one that we think most about. And the reason why is that it's probably one of the more widely expressed phosphodiesterases. It's in the most number of cells and tissues throughout the body. It has a really, really good KM, 1 to 4 micromolar. It's regulated by protein kinase A. It has numerous targeting domains. And although I'm not going to get into this, there's 4 different isoforms of PDE4. So these are the main isoforms, but then there's additional isoforms. So there's PDE4A, B, C, and D, and you'll see those nomenclature come up when we talk about the data, the preclinical and the clinical data for PDE4 inhibition. And then even though there's A, B, C, and D, there's splice variants within the A, B, C, and D. So the estimates is that there's somewhere around 110 different phosphodiesterase proteins that are functionally active in cells, localized to different areas of the cell, different cells, et cetera. All of which would be inhibited by phosphodiesterase inhibitors. PDE5, most people know as the target of Viagra. PDE7 is something that's more actively studied right now. It's a cyclic AMP specific PDE with an incredible KM. And there's a lot of research into developing inhibitors against PDE7. PDE8 also, very little is known about PDE8, but also expressed in numerous tissues with a very good KM. And so if you look on this figure, you can see different areas where PDEs are expressed. As I mentioned, PDE1 is in the brain, PDE2. You have different localizations for PDE4. PDE4 can be in the lung epithelium. That's why a lot of people develop PDE4 inhibitors for asthma and other lung biologies and lung pathologies. There's a whole host of the PDEs, 1 through 4 are in muscle. PDE4, 1, 3, 4, 5, and 11 are in vascular smooth muscle. So, and PDE4 is also in mast cells and other inflammatory cells, T cells, et cetera. So you get the idea that there's a variety of different localizations for these PDEs, and they obviously have different biologies in each location. Okay, so why did everyone get excited about the idea of blocking PDEs for inflammatory disease? Why were these drugs that we're going to talk about in a little bit developed? Well, the literature actually goes back many, many decades, okay? So all the way back into even the 70s, it was appreciated that cyclic AMP has effects on psoriasis and eczema. There is this famous story about Voorhees. I don't know if you guys know who he is, but I think he's in Michigan, is there? Yeah. Where he would get up and talk about psoriasis. And I actually wasn't there to see these lectures. This is literally like lore within the cyclic AMP field. They would get up and talk about psoriasis and cyclic AMP, and in the 1 year he'd get up there and go, psoriasis, they have a picture of psoriasis, they put cyclic AMP up, and then point to all the things psoriasis did. The next year, he would have the same exact slide, and cyclic AMP was down, and it was doing all the things. And then another year later, it would be like PKA. And the reason for this is that at the time, people really didn't know how to measure cyclic AMP. They were doing bulk tissue cyclic AMP measurements. They had no idea about the idea of microdomains or the fact that cyclic AMP is actually transient. It goes up and then down very quickly. And if you prevent it from going up, you have biology. But if you prevent it from coming down, you have different biology. So at the time, they were doing very crude experiments. And what was clear is that cyclic AMP changed. That was what was obvious. If you were looking at somebody who had lesional skin versus non-lesional skin, et cetera. So that much that much was clear, and that led to people like Bevo and others who started characterizing these phosphodiesterases, people who discovered these enzymes, and looking in the immune system and asking questions about what their role might be. And so very early experiments showed that PDE4 isoforms are widely expressed in inflammatory cells. Okay, so the enzyme's there, right? Maybe it's doing something. There were a variety of reports that show that decreased cyclic AMP, which was due to upregulated PD activity. Now, bear in mind, they weren't actually measuring the cyclic AMP, they were measuring the PD activity, and they were assuming the cyclic AMP was low because the PD activity was very high. In monocytes, what was present in monocytes for atopic dermatitis patients. So they took monocytes from normal patients and atopic derm patients, they measured PD activity, and it was higher in the atopic derm patients. And the supposition was that was driving the disease. Peripheral blood mononuclear leukocyte PD activity was found to be higher in atopic children as compared to controls. This was found in both the blood cells, it was also found in the tissue. So again, the supposition is that the elevated PD activity is driving the disease. And then, of course, cyclic AMP activation was found to be altered in involved versus non-involved skin. I sort of alluded to the stories that went back and forth on that. This is, if you summarize all that data, essentially that is what was consistently shown, that there was elevated cyclic AMP in the involved versus the non-involved skin. Okay, so that drove people to develop inhibitors to the phosphodiesterases to ask simple questions in the lab with mice and cells and say, can we perturb this system and have some efficacy towards these diseases? This is a sort of a summary of that data, and then I'll show you some more specific examples that are a little bit more recent. PDE4 again, and a lot of this I'm going to focus on PDE4 only because the drugs are against PDE4 that we're going to talk about, but there are other experiments that were done with other phosphodiesterase, other isoforms and drugs against other isoforms that also had positive preclinical work. But again, I'm going to focus on PDE4. So PDE4 knockout mice, where they knocked out the PDE4 gene in all of the cells, all the tissues of the mice, had reduced neutrophils in the blood. Right? Neutrophil level was just down. PDE4 knockout mice had decreased cell proliferation in response to LPS. For those of you who don't know what LPS is, it's a bacterial protein. It potently stimulates the immune system. You get a lot of T cell neutrophil recruitment to the site where you've injected it. You get a lot of proliferation in the blood. And you had decreased proliferation in response to LPS when you knocked out PDE4. PDE4 knockout mice were deficient in neutrophil migration to keratinocytes in skin disease models. So this got a little bit more sophisticated. We weren't just bulk stimulating the immune system. In these cases, the experiments were designed to specifically stimulate the epidermis and look at how the immune system trafficked to the epidermis. And what this showed was that there was a decrease in the migration when PDE4 was not functioning in these animals. Then inhibitors were developed, which were specific to PDE4. And this was found to reduce bacterial pathogen-induced inflammation, and all of these previous examples I mentioned were all recapitulated. So that told us that it wasn't just knocking out PDE4 and leading to some compensatory effect in the animal, that if we acutely inhibited it with a drug, it was possible to affect these biologies. So these are some examples. And as a pharmacologist, so my PhD is in pharmacology, so I'm very critical of these types of experiments, especially with preclinical models, drug company studies, clinical trial studies, because there's usually, there's an efficacy, but then there's not a lot of pharmacodynamic, pharmacokinetic studies to back it up. In the beginning of the PDE4 story, there was a lot of that, so that always made me very happy to see. This is an example of one of those experiments. So these are all just different inhibitors against PDE4A. So all these are just different drugs that block PDE4 at different potencies. The potency is on the x-axis. So really potent, not that potent. And then they looked at T cell inhibition. So the really potent ones were really good at blocking T cells, and the not so potent ones weren't so good at blocking T cells. You want to see something like that. That tells you that your drug has, at least in the context of this assay, high levels of specificity. They did this for PDE4D as well, and found a similar relationship. This is all in vitro, so then they wanted to look in vivo, and this is time after LPS treatment. I mentioned what LPS was and what it does, and it leads to both cellular proliferation as well as the release of inflammatory cytokines like TNF. And what this nicely showed is that if we knock out PDE4B or PDE4D, we have less proliferation of cells, and we have less cytokine release. So that told us that at least in an animal model with either a drug or a knockout model, we can prevent inflammatory reactions, not specific to atopic derm or psoriasis, but at least inflammatory reactions. There you go. Okay. All right, so here we're looking specifically at cytokine release. All right, so they wanted to go a little bit deeper in some of these papers. I've pulled some figures from different papers that I felt really highlighted what moved this field forward. Before we were looking mostly at T-cell proliferation. Now we're looking exclusively at cytokine release. So the same kind of a graph, really nice. I mean, it's almost a one-to-one direct correlation between the amount of drug and the inhibition of TNF-alpha. This is into in vitro systems with inhibitors to 4A and 4D. And then we have the genetic correlation. So in this case, we have LPS-induced cytokine release in the wild type, and you see there's a drastic reduction in the amount of cytokine that's released in response to LPS. Here's TNF-alpha. And then in this figure, what they did was they wanted to see if they could recapitulate what would be an activated system. So they used this drug called forskolin, which is abbreviated FSK. FSK potently stimulates TMACs, and they got, as you can see, a really nice reduction. And then they used rolopram, which is this inhibitor of PDE4, all PDE4s, and they got a further reduction. And that was important for them to see happen in the wild type animal. But then they look at the knockout animal, and they lose that PDE4 inhibitory effect. So that told them that the drug was most likely specific towards the PDE4. If you get an effect in a knockout with the drug that blocks the protein you knocked out, that's never a good thing. And this is just one example. There's many examples where they work tirelessly to show that these were in fact specific to the protein that they were inhibiting. And even in the case where there's drugs that can distinguish between PDE4D and PDE4A, they've done specific knockouts to those different isoforms to show these drugs are very specific. So these are really good drugs. They're really effective at hitting the target they say they're hitting, and they're unlikely to have off-target effects. Right? So that's the take-home message from this. And then all of those experiments I showed previously were all taking snapshots of a disease process, T-cell proliferation, cytokine release. This particular model actually looks at the ability of PDE4B knockout mice to prevent death due to high levels of LPS injection. If you inject enough LPS, you get massive inflammatory cytokine release, and the animals will perish. Not having PDE4B prevented the demise of those animals. So that says that you're having a drastic effect on the biological response of these animals, and you're preventing death or preventing inflammation leading to death when you don't have PDE4B. And there's many other examples where they did preclinical studies for atopic derm and psoriasis, but the take-home message was essentially the same. Okay. Are there any questions at this point? No, just soaking it up? Okay. All right. All right, so
I do have a question. Do you always, like when they're doing these kinds of tests, do they always have a knockout and compare the drug in the way that you suggested to show that it's really working?
No.
So that's
Do they, who's they?
Whoever they is that you've been saying they. In the testing of a preclinical testing of a drug, is it typical to have a knockout model to compare the drug's biological effects?
If you know the target of the drug,
if you know the target,
you should be doing that.
Okay.
And I think that in most cases they do, but a lot of cases drugs don't have targets.
Okay. So they just have an observed effect.
Yeah, a lot of the drugs we use, we didn't know the target before, like when they were released.
And other things that were
like aspirin,
Yeah.
And then later on, it was found what these drugs block. Biguanides are a great example. If you know what metformin is or fenformin, there's like 3 different potential targets of these drugs. So sometimes it's not even possible. But when you have a designed drug, which is designed to specifically block a given protein,
then you could design the knockout and
that should be a first proof of principle to show that your drug is specific. Otherwise, and I think it's easy enough now that most people do it, but it's not always done. And I encourage anyone who's looking at a drug where it either has limited efficacy or has a lot of off-target effects to look into the literature as to how it's actually used. because it's not always there. Sometimes it's anecdotal. Okay, so let's talk about the first FDA-approved inhibitor of PDE, and that's apremilast or Otezla. So this is the structure, and I know all you are looking at this and going, "Gosh, this looks like thalidomide." Right? Everyone's saying that. I know none of you are saying that. But that's actually how it was found. The history is that Celgene acquired the rights to thalidomide, and they started making thalidomide derivatives, and that has led to really great anti-inflammatory drugs, which some of you might use in the clinic. But they found that there were some derivatives of thalidomide that had other biological activities, and one of them was this drug, which is apremilast. And what they found was that this particular drug was incredibly good at blocking the activity of PDE4s, right? So it really didn't do anything for the PDE1s, 2s, 3s, 5, 6, 7, 8, 9, 10, 11, just didn't do much of anything. But as you can see, all the isoforms of PDE4 were incredibly sensitive to this drug, okay? And then they started doing experiments to look at what the ramifications of this drug were. This is sort of this is in vitro, and this is looking in vivo. And again, that's why they're comparing it to these other thalidomide derivatives. So this is percent inhibition. So this is a really good inhibitor. If you get to this point, you've inhibited all the PDE. It's sort of a weird graph to look at because the more it goes up is the more inhibition you have. So these don't inhibit much at all, and these are your pure thalidomide derivatives. And then you have roloprem, which is your prototypical PDE4 inhibitor, and then apremilast is right there. And then roflumilast is another PDE4 inhibitor, which has actually been used in the clinic for asthma and other inflammatory diseases of the lung, which is actually a better PDE4 inhibitor, but also has a lot of toxicities. So extending on those experiments, there was a comparison done between apremilast and, so here's lenalidomide, which is the drug that's one of the thalidomide derivatives that's prescribed, and other thalidomide derivatives. So this was to show that TNF-alpha is blocked pretty nicely by all three of them, right? We know the thalidomide derivatives are anti-inflammatory. And if you look at interferon alpha, then you get a really nice inhibition by apremilast, but not really by lenalidomide. If you look at IgG production, which would be your plasma cells, you get really no effect of apremilast, but you get a really nice inhibition by the thalidomide derivatives. And then if you're looking at IL-2 production, you actually get an increase with the thalidomide derivative, but you get an inhibition with apremilast. So you can see that even though these are all, the points of these figures were to show you that even though it's a thalidomide derivative, because of its unique PDE4 inhibitory activity, it has unique biological properties. And those experiments plus others led to a variety of clinical trials. I want to point this one out. I just think this is a really well-run clinical trial. There was a screening done and a randomization of 1 to 2, where they took people, either put them on placebo or apremilast 30 milligrams twice a day. And they did that for a period of time, 32 weeks. At 16 weeks, they took the placebo patients and they moved them onto apremilast. And then, depending on what their PASI scores were, they did other alterations to their dosing regimen. And then, in this particular group, if they had greater than a PASI 50, then they actually moved some to a placebo arm, which then they were allowed to move off once they had a loss of effect. So if you look at this data, the patients who are on apremilast, by week 16, which is usually a good benchmark time to evaluate for psoriasis drugs, you have about 50% of the patients reach a PASI 50, about 20 to 30% reached a PASI 75, and about 10% reached a PASI 90. And this was relatively well maintained over a period of 32 weeks. So the drug was fairly effective at treating psoriasis. When you look at their other arms, here, this is the placebo group, and at week 16, they were switched to apremilast, and you can see that they nicely achieve clearance up to the level of those who started on the drug 16 weeks earlier. Right? So a really nice proof of principle that this particular drug is effective at clearing psoriasis even after a placebo arm. So now we have mean percent change, right? So the mean percent change going down is good. It means you have a large percent change. And so this is looking at it in different directions. We're not looking at PASI, we're looking at percent change of the PASI score. And the same thing, if you're on placebo, not much is going on. Then you go on drug and you reach the same level as those who were on the drug from the get-go. And this is, again, mean change in PASI. This is based mean change in pruritus VAS score. So just another indicator of drug success. In psoriasis, you have basically PASI, VAS, a few others, global assessment scores, and then we'll talk about some of the other scores for atopic dermatitis drugs in a second. Okay, so one of the things that happened, so that's the trial that led to the FDA approval, right? So it's clearly has efficacy. Side effect-wise, fairly minimal. A lot of times we talk about it as a non-immunosuppressive drug. I don't like that term. It is immunosuppressive. It does suppress the immune system. It just doesn't do so to the extent of the other immunosuppressive medications, and the FDA felt that blood monitoring wasn't necessary. And that's probably true. It doesn't block the immune system enough to necessitate blood monitoring, but it is blocking the immune system. So I don't like to tell my patients that it's a non-immunosuppressive medicine. It just doesn't work by the traditional immunosuppressive mechanisms like steroids, et cetera. The main side effects for those of you who put patients on the drug are GI upset. And I was talking to Adam about this before. A lot of time it's like watery diarrhea. Does anybody want to venture a guess as to why? This was not surprising to me when this side effect came out. Anybody want to venture a guess as to why a PDE4 inhibitor would give you watery diarrhea?
Do the Cyclic AMP going water? Like, there's not a lot of toxins from the bacteria that cause diarrhea.
Like what?
Coli.
Cholera. So cholera toxin functions to ADP ribosylate, the heterotrimeric G protein that you guys are all now experts on, which activate that TMAC. So the cholera toxin functions to make that GS alpha always on. It's always functioning. It's always making cyclic AMP from the TMAC in the gut. That's how cholera toxin. That is what cholera toxin does. The phosphodiesterase is going to raise cyclic AMP in those people who have activity at those transmembrane adenylate cyclases in the gut, and that's why not everyone gets GI upset, but some people do. That's why some people get used to the drug after a period of time. Most likely what happens in those patients is they tune down, they turn off the TMAC activity that's going on in the gut that's leading to the watery diarrhea, and some people just don't. And that's it's hard to prove these things, but that's most likely the reason why some people will get used to the drug after a while and some people won't. So talking about other applications for apremilast, it's been shown to be highly effective for psoriatic arthritis, nail psoriasis. There was 60% improvement in 4 months. Scalp psoriasis had about a 50% improvement at 8 months. Palmoplantar psoriasis, 69% improvement at 8 months. I do know that, at least in my clinic, when you have somebody who has isolated scalp psoriasis, not really amenable to narrowband UVB, this is a good choice. I have cleared some people using it. Even palmoplantar psoriasis. I think we talked about one patient that you used it for, yeah. So, when it works, it works. And in areas where you don't have a lot of other options, or you don't want to put somebody on Cellcept or methotrexate or some other biologic, or they're not amenable to biologics personally or for some other reason, it's a really good choice for psoriasis. Okay. Any questions about apremilast? Because then we're going to switch to you, Chris.
On the label regarding side effects, on the label for apremilast, it doesn't mention that depression is a possible adverse event. Can you comment anything on that, or actually want to? PDE4 is present in many cells throughout the body. It's in the brain. It's in nerves, it's in the gut, it's in the liver. So all these different side effects, I can create a scenario. I don't have any hard data, and I don't think there really is data to really nail down why that happens. But it doesn't surprise me that a drug that hits PDE4, but not specifically in a given cell type, could lead to these side effects. The weight loss is another thing. About 10% of people get weight loss, right? Not such a bad thing. What's interesting is that the TMACs, if you increase their activity, you get weight gain, okay? Because this responds to glucagon and other things. But the cyclases that we study, the soluble AC, in our knockout mice, okay, they have a 10% weight gain, right? So it's the opposite. So we knock out, we lower our cyclic AMP from our cyclase, we get weight gain. And when you increase cyclic AMP from the PDE4, you get weight loss. But the TMAC biology is the opposite of what you'd expect. So this is an example of where this is a microdomain that we're actively studying because we think that the soluble AC microdomain with a PDE4 is controlling weight, right? So we have drugs that can affect the soluble AC different from the TMACs, and this is the ways in which you can start to tease this system apart, use combination therapies to really hopefully reduce certain symptoms and maybe increase the efficacy of what we want. Okay, so crisaborole or Eucrisa. So this is a really interesting molecule. So there's a company, it was called Annacore, the history of this drug. So as a pharmacologist, I like to know the history of all the drugs. So Annacore was this company, and it's really, if you want to look up the history of Annacore, it's a really good story. It's a guy named Lee Zane. He's a dermatologist out of Stanford, really, really nice guy, who a few years after finishing his residency, moved to Annacore and became their chief medical officer. This was like a nothing company. It was really small, but they had a really interesting platform. They were able to synthesize chemicals incorporating a boron atom. And boron is a really interesting atom. Because of its chemistry, it increases the penetration of drugs. So drugs made with boron are able to penetrate more deeply into the skin than drugs made with other molecules. So chemists developed this platform to make chemicals with boron, right? That was the basic idea. And they made a ton of chemicals. And Lee guided what they made chemicals for. So they had an antifungal that was FDA approved, okay? And then soon after, Eucrisa. So the reason why it's a good story to tell you guys if you're interested in industry is that Annacore, as you may or may not know, was just acquired from Pfizer for $6.5 billion. So Lee made a good bet. But it's a good story because it's a chemical platform guided by a smart dermatologist who knew what drugs, what targets needed to be addressed, and again, a chemical platform that could be used using crystal structure and targeted design to develop really good, smart drugs. And they're developing a lot of really great drugs using this platform, not just in dermatology. So they came across this particular molecule, which was called AN2728. And this is looking at percent inhibition of all the different PDEs. And if you look at PDE4, it had 99% inhibition. So basically obliterated the activity of PDE4. But it didn't obliterate, it did affect other PDEs, PDE1, PDE2A, PDE3. So it definitely is not, unlike apremilast, where apremilast seemed to really not affect any PDE except PDE4. The data with AN2728 suggested that it did affect other phosphodiesterases. They took it to the next level, which was look at cytokine release, and they showed that there are specific cytokines that were very potently inhibited, like interferon, IL-2, and then there are other cytokines that really weren't inhibited, like IL-1 beta. Right? So there was a cytokine profile change that was induced by this particular molecule. And then this is relatively new, so it was just FDA approved. And this is data from the study on that initial FDA approval. So what this uses is a scale called ISGA, and I put it down here, Investigators Static Global Assessment. And what this attempts to do is to create these criteria for disease involvement, like how bad the disease is. So scale of 4 is considered severe, and this is deep or bright red erythematous and severe indurated, I can't read this one, population with oozing and crusting. And then there's obviously there's a whole transition all the way down to 0, which is basically clear skin. And so what you can do is you can enroll patients who fall into category 4, and then you can mark the ability of this drug to bring them to category 1 or category 0. And that's essentially what this graph is showing. So The comparison was between crisaborole and vehicle. And when you look at vehicle, as we all know with our atopics, vehicle does have some efficacy. And this is the efficacy of vehicle application over a period of about 30 days on the success of the ISGA percentage. And so this is moving from 4 to 0 or 1. And you can see that about 25% of the patients had success. Whereas if you look at crisaborole, you get about 40% having success. So it's about a doubling of those who achieve this ISGA improvement of 0 to 1. Now that's just looking at the clinical presentation of the disease. What they did is they looked a little bit closer. So we're going to go into specific aspects. This is just looking at the percentage of improvement. So 32% to 25%, 31% to 18% with the ISGA. And then again, this is looking at the number of cleared 0 to 1. So went from 51 versus 42, 41 versus 29. So in each case, there was an increase in clearance. So sorry, I transitioned a little too early. I apologize. But then they looked at other aspects of the drug's efficacy. And one thing they looked at was pruritus. And as we all know, with atopic dermatitis, we call it the itch that rashes a lot of the time. So the itching, people staying up all night, scratching, these are some of the comorbidities that we associate with atopic dermatitis. And so one of their arms, and it's interesting because this arm, or this evaluation is essentially being utilized by every other PDE4 that's in preclinical. It's in clinical trials right now. They're all looking at pruritus because it appears that the phosphodiesterase inhibitors are quite effective at blocking pruritus, reducing pruritus. And so this is an example. So different days, the vehicle is in yellow and the drug is in blue. And pretty much one of the main positives that the patients reported when they were on the drug was that they felt less itchy, and the itch relief was almost immediate. It was really within a few days. There's other companies. I don't want to go into too much of the preclinical stuff because not all of it's publicly available. But there's other drugs that have shown that sometimes the pruritus is the reduction in pruritus is seen at 48 hours, really, really, really fast. And this is showing what the disease looks like when you're at baseline versus at day 28. So clear improvement. And this is just to show you what the severity of pruritus scale is how it's used. So severe, moderate, mild, and none. And so there we're looking again at this reduction in severe. They're looking for a score of 0 or 1 with a greater than or equal to 1 grade reduction, right? So most of these people started severe and they wanted them to at least progress to 0 or 1. And it was very effective. So this is a bar graph just dictating other factors of atopic dermatitis that they evaluated. So erythema, exudation, excoriation, induration, lichenification, and in all instances, the drug was more effective than vehicle at reducing these symptoms of atopic dermatitis. So it was clearly had efficacy. The safety profile was really, really good. There really wasn't any of the skin thinning and other issues that we worry about with topical steroids. There is some burning. We were talking about this earlier. And this, it's possible that this is also elevations of cyclic AMP in the nerve cells or release of inflammatory cytokines due to cyclic AMP that are stimulating the nerve. It's really unclear as to why that occurs. Okay, so the data I've shown so far is how these drugs came to be, what led to people developing them as therapeutics, and the data that the FDA used to lead to their approval. Okay, so the question that I get asked a lot, and some of you might get asked as well when you prescribe these drugs, is why do they have limited efficacy, right? And when I say limited, I mean they clearly have efficacy. They clearly lead to a reduction in atopic dermatitis, a reduction in psoriasis. But if you're going to compare it to what we think of as systemic medications, it's limited. And just to give you an example, this is Humira, Stelara, and Taltz. These are the PASI 75s at 16 weeks, and it's 70 to 90, and 70 to 80. So you have to compare that to the positive 75 of apremilast, which is almost 30% at week 16, right? So it's not as effective as these other medications. So why would that be? Can we make it better, right? So we shouldn't just stop. We shouldn't rest on our laurels. Clobetasol. So ISGA is not used a lot in clinical trials. The only thing I could find was clobetasol, which had a 59% ISGA, comparing that to around 40%. So, whatever you want to compare it to, they're limited compared to our standard of care. Now, granted, they don't have a lot of the side effects and worries of the other medications that have far higher efficacy, but we can still try to leverage that. So why do they have limited efficacy? Well, the first thing is that they target, in the case of apremilast, which is very, very effective at targeting PDE4 and only PDE4. Eucrisa targets other PDEs, but let's just focus on PDE4 for 1 second. As I mentioned earlier, there are 4 different types of PDE4. There's A, B, C, and D. And there's splice variants of all those PDE4s. And they're in multiple different localizations, multiple different cells, and they're involved in multiple different pathways. So when you put a drug on the skin or you ingest a drug and it goes systemic, it's going to elevate cyclic AMP at all these different locations in all these different ways, activating to some extent many of these pathways, some of which might counteract the efficacy of the drug in the specific cells where you're trying to achieve efficacy. I showed you the preclinical data. They really are effective at blocking T cell cytokine release, T cell proliferation. They're very effective in those particular cells. But that might be mitigated by their effects outside of those cells. So I bring you back to this figure I showed you in the beginning. So go ahead.
Just a quick question. So you were talking about, I think, your linear regression model of T cell inhibition, right?
Yeah.
Based on, I think it was dosing of the PDE.
That's right.
So we know that different subtypes of T cells are upregulated in different inflammatory diseases. So have we thought about differentiating those subtypes, not just overall downregulation of the T cells?
So have we thought of it? Yes. Is it known for these drugs to some degree? There is very good data that's been published that shows that the Th17 cytokine profile is reduced when you give apremilast to patients. We don't know where those T cells are, and we don't know if it's because it's directly inhibiting Th17 cells specifically, or it's inhibiting some other cell that's leading to Th17 downregulation. But suffice to say, cytokines that are a measurement of psoriasis, Th17 cytokines, are reduced, right? Which makes sense because the disease is reduced. We just don't know what else is going on. And you make a very good point. There's multiple Th subsets. There's Treg cells. All of these have phosphodiesterases in them, right? Yeah. So, and again, that gets back to the cyclase, right? So excellent transition to this slide. So let's just make an example. Let's say this is the PDE4 that we need to inhibit in order to get an anti-inflammatory effect. So we need this cyclase to be activated, but we don't want these other cyclases to be activated. So why can't we just include an inhibitor to these other cyclases to prevent them, which also have PDE4s associated with them, but prevent them from functioning, so we only get cyclic AMP elevation at the microdomain, which we know is contributing to the T cell phenotype that we want. That's what my lab does. Right? So that's exactly what we use, genetic and pharmacologic reagents, to tease out these microdomains, to ask those questions, and to improve the efficacy of these drugs and better understand which PDEs are playing a dominant role by doing things like isolating T cells from different animals, crossing them, et cetera. But just exploring how these drugs work and in what context, I think, will illuminate these different scenarios and help us understand the side effects that we like 10% weight loss, and the side effects we don't like GI upset. And the reason why this is important, and we've sort of already alluded to this, is if you think about different diseases, and psoriasis is very well worked out, so I threw that one up there. These are all the inflammatory cells and non-inflammatory cells that we know have a role in psoriasis, right? So if you are aware of Nicole Ward's work, she showed that if you sever the nerve leading to a certain area of a mouse, you no longer get psoriasis in that area, right? And so you can use things like Botox to reduce nerve impulses to an area, and you don't get psoriasis. So there's a nerve component. The nerves have to be functioning. We know of pathergy, Right? We know if we irritate the nerves, we get psoriasis. There's models where keratinocytes are activated using JAK and STAT pathways, and that's sufficient to induce a psoriasis-like model in mice, where the immune cells are not directly stimulated, the keratinocytes are directly stimulated. So we know all these different cells are playing a role. And then if we ask, what does cyclic AMP or PDE4 do, you get a whole myriad of responses, some that would be counterproductive to what we're trying to achieve. So I'm only mentioning a couple of examples, but dendritic cells, right? Pretty important cells. They basically wake the T cells up and drive them towards a specific process. Well, if you inhibit PDE4 in these cells, this is all published stuff, you actually lead to an increase in IL-17 production and Th17 polarization with a reduction in Th1 polarization. That doesn't sound like something we want to do if we're going to stop psoriasis, right? This absolutely happens in mice, in cells, published work. But at the same time, those drugs are reducing IL-17 secretion and the proliferation of those Th17 cells. So it's kind of fighting against itself in a way, right? And so it really depends on where the patient is and the polarization state and when you hit them, and maybe what other medications you would want to package with the PDE4 inhibitor. Really, nothing's known about NKT cells, so it's ripe for investigation. Endothelial cells, PDE4 inhibition blocks cell influx, so this sounds like a good thing. We don't get cells into the tissue, so that's a positive. Keratinocytes, cyclic AMP leads to an inhibition of proliferation and cytokine release, right? So that's good. We don't want the keratinocytes to proliferate. I will tell you that's the published result, but in our laboratory, so spoiler alert, unpublished work, if it's soluble adenylyl cyclase, it actually is the opposite. So soluble adenylyl cyclase is controlling the keratinocytes in a way whereby if you raise cyclic AMP next to that cyclase, you would counteract the effect of the PDE4 inhibitor. And then you have fibroblasts. So PDE4 inhibition reduces fibroblast migration and activation. Again, sounds like a good thing. But again, these are all anecdotal studies that have been published, and they're not really well worked out, especially not with the drugs that are FDA approved. These are all with the old style drugs that weren't as specific as the ones that are FDA approved. Okay, so in summary, hopefully, I've proven to you that cyclic AMP is generated distinct microdomains, so we shouldn't think about cyclic AMP as just floating around in the cell, bags of cyclic AMP, and that these different microdomains have very different effects even within the same cell. The PDEs define these microdomains using different mechanisms. The PDE activity is altered in inflammatory skin diseases. The inhibition of PDEs leads to reduced cytokine release and T cell activation. That is absolutely true. That's why the drugs work. That's why there's efficacy. That's why they were FDA approved. They are effective drugs. And they are effective at treating psoriasis and atopic derm, but other inflammatory diseases. People have started looking at them in asthma and in other inflammatory lung diseases. They've looked at them in other types of like lupus. People are looking at them in all different types of inflammatory skin diseases. And I'm sure there's going to be some level of efficacy in some of those diseases. But because of a lack of PDE4 inhibitor efficacy, meaning phenotypic efficacy, disease treatment efficacy, is likely due to competing microdomains in the cell, not that the drug is a bad drug. The drugs are really good. As I showed you earlier, they hit the target, they block the target, there's efficacy from blocking that target. It's just that target's not only in the cell that we're trying to block, and we don't understand as much as we'd like to about which domains are important, and that's why I believe there's limited efficacy in the results we're seeing as when you compare it to other biologics and systemic therapies. So with that, I'll close. If there's any questions, happy to take them.
Nothing with the word yes. Being the first year, I've never used Influenza or Tesla yet, but I just wonder, has there been any signal in the surveillance in regard to melanoma because of PDE5? I don't know if the biology of PDE4 and PDE5 signaling is similar enough that we would see that.
So PDE5 degrades cyclic GMP. It's a good question. So just to remind everyone, PDE5 is exclusively cyclic GMP. PDE4 is exclusively cyclic AMP. That being said, there is a plethora of data talking about the role of cyclic AMP in melanoma. Everything I would tell you would be unpublished from my lab, so I don't want to just sort of throw unpublished data at you, but I'll just say that cyclic AMP, depending on where it's made, can affect melanoma differently. So some sources of cyclic AMP can affect resistance to melanoma. Some sources of cyclic AMP can affect how effectively melanoma develops in a mouse model. And those are not the same necessarily. So could it? It might. I've never personally tested it. When you test drugs that are FDA approved, they have to be under explicit instruction from the company that provides it. So all of my work is focused exclusively on inflammatory skin diseases. I guess you could use something like Rolapram or a non-specific PD4 inhibitor to answer those questions, but I haven't done it, and I'm not aware of anybody who has. Now, going to the cyclic GMP, I encourage you to review that literature yourself and be unimpressed by its strength. By its strength. I really encourage you to look at those epidemiological studies, read the papers, not that many of them, before talking to your patients about the use of PDE5 inhibitors. The best one point I'll give you is that there is no correlation with the amount of PDE5 inhibitor used and the incidence of melanoma in those patients. I'll leave you with that one piece of information. And the other piece, one other piece, when you talk about a drug that drives a disease that leads to no permanent effect in the cell, ask yourself how that's possible. For example, we all know about SCCs that develop when people are on BRAF inhibitors. Why does that happen? It happens because the BRAF inhibitor binds to wild-type BRAF, and in the context of a RAS mutation, KRAS, most likely in SCCs, you get this paradoxical hyperactivation of the MAP kinase pathway, and that is only because it's high levels of the BRAF inhibitor binding to wild-type BRAF with a RAS mutation, which is one of the resistance mechanisms in melanoma. So those patients develop SCCs. What happens when you take the BRAF inhibitor away? SCCs melt away. Right? The tumor is dependent on the presence of the drug. All those studies were people who took like Viagra like 10 years ago. Right? The drug's not hanging around. Now, that's different if you're talking about a drug that causes DNA mutation. Right? Different story. If the drug causes a permanent modification to the cell, then we're talking. Then cancer-inducing properties are different. But these are not those drugs. These are not permanent tonic effects that you get. And so once you remove the drug, I would expect all of those diseases to wash away.
I think the point is it's correlation at best, not causation. And I think both of those studies were from the same at best. And both of those studies were the same group too. It was the same.
There's a couple of groups. The epidemiology. There's some that say it does and some that say it doesn't. And I encourage you to read it. There's not that many papers, so I encourage you to read them. And then if you look at the biochemistry studies, there's only 2 papers. And really, really critically read those papers and ask yourself if it proves that Viagra would cause melanoma, right? And you can email me if you'd like to debate it over.
Right. I think we're going to end there just in the interest of time. So thank you, Dr. Zippin, for joining us.